It's a pleasure to have Maurice with us this afternoon, who will talk to us about supercharging CHO cell productivity using 2G, UNic technology, and Transposon innovation. The floor is yours. 10 minutes.
Thank you. So, welcome to the presentation of ProteoNic's 2G UNic Transposon technology. In this presentation, I'll introduce 2G UNic technology to you, and then move to our recently launched this meeting, 2G UNic Transposon technology, which is now available for licensing. So 2G UNic vector technology is a patented technology. It's been around for quite a while, with pharmaceutical companies using it in their cell line development processes, with cell lines in clinical development. It's used by CDMOs as well. You may have seen Abzina's presentation yesterday, and commercialized through licensing. So at the core of our 2G UNic vectors is the 2G UNic elements, comprising of a double promoter system built around a human CMV promoter, which is functioning as an intronic promoter to the first promoter. So the first promoter adds epigenetic regulation, so basically preventing epigenetic downregulation, making expression more or less independent of the integration site in the genome. So together guaranteeing high mRNA transcription. Added to that, we have an optimized 5' UTR sequence that adds improved ribosome affinities, optimized mRNA transport and stability, resulting in more protein per mRNA molecule. And together, these result in an increased protein expression per integrated copy in the genome. Technology can be applied in different ways. We can introduce it in your existing vector system. Or alternatively, we have complete vectors where you just need to insert your gene of interest, either single gene or bicistronic. Using this vector will immediately result in increased titers in stable bulk pools or mini pools, and from these bulk pools you can, isolate clonal cells with significantly increased titers. Basically, no process change is required compared to your current work stream. We have shown that 2G UNic increases expression in different cell lines. All standardly used CHO cells we have worked with different selection markers, antibiotic markers, GS, DHFR, and for increased expression of different types of molecules, ranging from well-behaving antibodies to Fc fusion proteins, bispecifics, trispecifics. And we see that these increased yields are obtained without loss of cell line stability. So cell lines are stable, process is scalable, and without change of product quality compared to reference vectors. Here's an example. It's a CDMO project expressing a bispecific antibody in CHO source cells. On the left hand, in the gray bars, you can see the competitor vector technology with only one clone reaching about 2 grams per liter. With 2G UNic technologies, clones reached up to 5 grams per liter, and many more high-producing clones were obtained. So that was 5 grams per liter. If you have a well-behaving molecule and process that supports high-level expression, the technology can also be used to achieve titers well over 10 grams per liter. So this is a targeted integration slide showing, this is data from a client having their own targeted integration cell line producing polyclonal IgG. With our standard vector technology, they obtained about 0.5 gram per liter in a fed batch process. With 2G UNic elements, they increased this to 2 grams per liter, and with a little bit of process development, up to 3 grams per liter. For a single copy, I think these are pretty good titers, showing that you don't need many copies. You need active copies. And that's exactly what ProteoNic's vector technology can help you with. Clients have used the technology in combination with different transposon technology. And we have seen from those evaluation studies that they combine the best of both technologies, giving the high expression per copy from the 2G UNic elements and the efficient genome insertion of complete copies by the transposon. So we're excited to announce that we now have our own transposon, the 2G UNic Transposon, that is giving all the benefits of both technologies. The transposon vector is just a standard 2G UNic vector equipped with inverted terminal repeat sequences. And we immediately saw that there was an increase in expression in stable bulk pools. So this is trastuzumab expression in the chosen cells. In green is the 2G UNic vector, so this is random integration. In magenta is the 2G UNic transposon, showing a twofold increased titers in its bulk pools. And we see similar results with other molecules. So here's expression for etanercept, trastuzumab, and a bispecific antibody. So this is a two-vector system. I mentioned that 2G UNic vectors can also be used with other transposons. What you see here is the transposon alone that we're using in pink. So the first bar, it's not expressing very well. Piggyback alone is expressing a little bit better. 2G UNic random integration shown in green here. And the last two bars in magenta, the 2G UNic transposon, and in darker yellow, the 2G UNic vector with piggyback. So you see the max titers in these bulk pools are very similar for the two transposon technologies. If we look at individual cells in the bulk pools, we see that there's an increase in the percentage of high expressing cells. So here on the top, on the left, you see the 2G UNic vector. About 3 to 5% of the cells are high expressing. And on the right is after adding the transposon, you see that this is increased to about 20% of the cells. So five-fold increase in high expressing cells. And we're currently in some evaluation studies with different companies. We see that the increased titers in the clones are confirmed now. Unfortunately, I'm not able to share this data yet. In addition to the titer, what we also see with transposon is that selection is significantly faster than the random integration process. So in green again, the 2G UNic vector in our chosen cells, it takes approximately 3 weeks to complete selection, and there's a significant drop in viability. The transposon process is much more efficient, resulting only in a minor drop and completed within 2 weeks. So in summary, the 2G UNic transposon is giving faster pool recovery, increased titers. It's compatible with different transposon systems. So if you have a transposon system already and you would like to boost your titers, then you could add 2G UNic technology. Or if you don't have access to any of those technologies, we're happy to license the 2G UNic transposon technology. So the QR code is leading to our webpage where the presentation is also available. And I have 45 seconds left, so if you have any questions, please let me know after the meeting. Or Jonathan and Shell are also here to discuss license options. Thank you.